Hi, it's Mr. Anderson, and in this video I'm going to talk about cells. Life is made of cells. It's the basic unit of structure and function. And also, all cells we have must come from pre-existing cells. And so how does that work? It's through a process called cell division. This is going to occur in all cells, both prokaryotic like bacteria and eukaryotic like you. And through cell division, one cell can become 2, which can quickly become 4, and then 8, and then 16, and so we get exponential growth in cells. And to give you an idea how quickly this goes, you started life as one fertilized egg, or one zygote. But those were quickly dividing over time, and it eventually created an adult that's going to have around 10 trillion cells inside your body. And they all came from that original cell. Now, to even increase that number quite a bit, just look in your gut, and we're going to find that the bacteria living in your digestive tract are going to outnumber the cells in your whole body 10 to 1. There are going to be 100 trillion bacteria just living in your gut, and they're going to be outnumbered about 10 to 1 by viruses living in your gut. So we're going to have quadrillion viruses living in your gut. Now, viruses aren't technically cells, but everything else I've described here is going to be reproducing, or it's going to be created through a process called cell division. And there are really 3 types of cell division. In prokaryotic cells, or in bacteria, they're simply going to divide through a process called binary fission. That's where one cell, one bacteria cell, is going to make 2 cells that are exact copies of the original cell. In eukaryotic cells, we have something similar to this. We're going to have cell division, or we sometimes just refer to it as mitosis. Technically, mitosis is going to be division of the nuclei, and then we're going to have cytokinesis, which is division of the rest of the cell. But we generally just say mitosis is when one eukaryotic cell makes 2 cells that are exact copies of that cell. It's a lot like binary fission. And then the third type of cell division is going to be meiosis. Meiosis is going to be when we create sex cells, or when we create gametes. Mitosis and binary fission ensure that we have exact duplicates of that original cell. Meiosis is totally different. Meiosis is going to make cells that are different. They're going to have half the genetic information, and they're going to give us variability. And so in us, sex is important because it gives us variability in the next generation. In bacteria, mutation, and there are some other ways that they can share information, is more important. So let's start by talking about bacteria. They reproduce through a process called binary fission. In this, we're going to have this adult cell, so we're going to have this parent cell. It's going to be made up of, you can see here, ribosomes. It's going to have a plasmid here, which is a little bit of extra DNA. But its genome, or all of its DNA, is going to be wrapped up into one loop. And so what do we want to do in binary fission? We want to take one cell, and we want to make 2 cells that are identical to that. And so the process is pretty simple. What the bacteria cell starts doing is duplicating all of its DNA. So the first thing it'll do is DNA polymerase goes around the whole loop, and it's going to make an exact copy of all the DNA in the bacteria. One thing not shown in here is that we're also going to duplicate our plasmids, and we're also going to have, ribosomes increasing as we increase the number of DNA as well. What happens next is it's going to be pulled to the side of that cell, and really what's going to happen is the DNA will actually attach to the cell wall, and then eventually that cell wall is going to split in half, and we're going to have 2 bacteria cells. How are they different? They're not really different. They're exactly the same. The DNA in this cell and the DNA in this cell is going to be exactly the same. And when we divide in half, we're going to divide in half those ribosomes and the plasmids as well. And so again, bacteria don't do mitosis. They don't have sex. They simply reproduce through a binary fission. And this happens incredibly quickly. So in a typical E. coli, they can copy themselves, make 2 copies of one cell in 20 minutes, which is just crazy how fast that occurs. But again, we don't go through binary fission because we're eukaryotic cells. And before we talk about mitosis and meiosis, we should make sure you know the difference between what a gamete is and then a somatic cell. And so eukaryotic cells are going to be everything from the chimpanzee to the bees, to the flowers, to the mushroom, to the yeast. They all have a nuclei. And so there are 2 types of cells that we find inside eukaryotic organisms. First are going to be the somatic cells. Somatic cells are going to be the body cells in your body. And then we're going to have the gametes, which are going to be the sex cells. So in humans, the only cells inside our body that are going to be gametes are going to be sperm in males, and then the eggs or the ovum that are found inside females. How are they different? Remember, somatic cells, or the body cells, are going to be diploid in humans. They're going to have 2 copies of every gene and therefore 2 copies of every chromosome. Gametes are only going to have 1. So sperm and egg each provide you with half the DNA that you get. So how do we duplicate a typical cell? Well, first we should say, when do we do that? Well, remember, when you go from a zygote to an adult, we're doubling the cells over and over and over again. And then we're constantly replacing the cells in our body. So as cells become worn out or damaged or removed, we have to replace those with other cells. And we do that through mitosis. Again, what is mitosis? It's technically the division of the nuclei, but then we're going to have division of the rest of the cell, which is called cytokinesis. So what do we start with? And we're going to make this real simple. This is a simple cell. It's a diploid cell. It's 2n equals 2. That means they have 2 chromosomes. These 2 chromosomes are going to be homologous. That means that they're the same length. The reason why they're different colors is you get one from one parent and one from another. And so in mitosis, let's look first, before we talk about the specifics of what we're going to get out of mitosis. We're going to start with one cell looks like this, and we're going to end up with 2 cells that look exactly like that first cell. So in mitosis, we want to duplicate the cell. And so let's watch that. What's the first thing that happens is we have to duplicate all of the DNA. And this occurs during interphase. What's going to happen is this chromosome right here is going to copy all of its DNA. And now we're going to have what are called sister chromatids. And so we have a chromosome on this side, a chromosome on this side, and they're exactly the same. So they're an exact duplicate of that original chromosome. And you can see that we're going to have that on the other homologous chromosome as well. What happens next? They're going to meet in the middle. They're going to line up. These lines represent a spindle which is going to attach to either side, each of those sister chromatids, and then they're going to be pulled apart. They're going to be pulled apart. And eventually, so this would be the mitosis when we divide the nuclei, and then the whole cell is going to split in half. We're going to divide that cytoplasm, and that's called cytokinesis. And so what do we end up with? We end up with 2 cells that are both diploid. Our original cell was diploid as well. So let's take a look at what this really looks like. We're looking right here at cells that are undergoing mitosis, and so they're growing on a growth medium. And if we just play the video, this is time lapse, and so it's speeding it up quite a bit. But what will happen if we look at like this cell right down here is that you can see it kind of come together, and the nuclei is dividing up the genetic information, and then the rest of the cell is going to divide. What do we end up with when we're done with that? We have 2 identical cells. And those are going to grow, and then they're eventually going to divide the nuclei, and they're going to grow, and they're going to grow, and they're going to grow. And cells, unless they're cancerous cells, are going to keep growing on here until they fill up this whole medium. But we need to talk about meiosis, which is the third type of cell division. What happens in meiosis? Meiosis is when you take one cell, and we're going to make 4 cells out of it. Now let's look how this is a little different than in mitosis. In mitosis, remember, we took 1 cell and we made 2 cells. In meiosis, we're actually going to have another division. But there's something even different that we didn't see in mitosis. Remember, in mitosis, we want to make sure that each of those cells that we make are identical to that original cell. You can see here that the 4 cells that we create at the end are going to be totally different than that original cell. And they're going to have half the genetic information. Remember, I said the sperm and the egg have half the information that's found in the original cell. And so what's going to happen? Well, let's watch how this begins. First thing that happens is you're going to duplicate each of these chromosomes. And so we're going to make copies of the chromosomes, but then something odd happens. What happens that's odd? We're going to take a little bit, in this case, we're going to take a little bit of this white chromosome here, and it's going to cross over with this red chromosome right here. So we're actually swapping chromosomes. And so that process is called crossing over, but what you're really doing is you're crossing over, or you're making a new chromosome. It's a combination of the chromosome you got from your mom and your dad. It's a chromosome that's never been built before. So those will eventually line up in the middle, and then they're going to divide, and then they're going to divide again. And so they'll divide in half. So this is meiosis 1, and then they'll divide in half again, and that's called meiosis 2. So what do we create when we're done? We're going to create daughter cells, but each of those daughter cells are going to be different. Each of these would create a new sperm or a new egg. Now it's going to be much more complicated in us. In humans, remember, we're not going to start with 2n equals 4, with 4 total chromosomes here. We're going to start with 2n equals 46, and so we're going to start with 46 chromosomes. So it's going to be really, really different when you get to the end. And so we can create literally billions and billions of different types of cells through meiosis. And so that's going to give us variation. So to summarize, what do we have? We have 3 types of cell division. We have binary fission. That's going to be found just in bacteria, where you take one bacteria, first copy the DNA, and then the rest of the cell is going to divide through cytokinesis. Each of these daughter cells is going to be identical to that first cell. If you're looking at eukaryotic cells, we've got mitosis. What happens in mitosis? We start with a diploid cell. So here the reds and the greens are each going to come from different parents. So we have 4 chromosomes. They're going to duplicate first during S phase of interphase. They'll line up in the middle and they'll be pulled to the side. Now, if we look here, these two cells that are produced in mitosis are identical to that original cell. And that's what mitosis does. It makes exact copies. If we start with meiosis, meiosis is going to start with an original cell that looks very similar. But instead of lining up like this, where we separate the sister chromatids, they're going to line up with the homologous chromosomes wrapping around each other and swapping bits of that chromosome. They'll divide in half, and then they're going to divide in half again. And what we're going to create is gametes. Those are going to be sperm and egg that have half the genetic information and a new combination of that genetic information. So again, this will be fertilized by a different sperm, and we get back to a zygote. Where do we go from the zygote? We go right back here to mitosis, where we can copy that cell over and over and over and over again. And so that's cell division. It's found in all cells on our planet. It shows us how one cell can go to all the cells that we have today. A good question might be, where did that original cell come from? But that's for another podcast, and I hope that was helpful.